Hello students, in this video we will discuss about some important organic compounds like amino acids, lipids, etc. Amino acid is what when you have studied a topic of chemistry or when you gone through with the study of protein, then you know what is protein. When various type of amino acid bound together with the help of peptide bond, then form that form polymer of that amino acid is further referred as protein. So what is protein? What are their structure? What are their types? We'll discuss later on. In this video, we will discuss what is amino acid. Amino acid, guys, further comprises four functional groups. One is hydrogen, one is amino group, one is hydrogen group, one is amino group, one is acidic carboxylic group, and one most important R group. This R group actually a functional group that is variable. Variable means what? Sometimes this R group is substituted by hydrogen, sometimes it is substituted by methane, or sometimes this R group is substituted by hydroxy methyl group. So guys, I would like to mention over here that the physical and chemical properties of amino acids are based on these four groups, especially when R group is substituted by hydrogen, methane, or hydroxymethyl, then the physical and chemical property of amino acids are also get changed. You know, uh, like in English alphabet, there are 26 letters, and when these 26 letters combine in various patterns, then they form a huge number of words. In the same way, there are 20 type of proteinaceous amino acids. There are 20 type of proteinaceous amino acids. When these 20 type of proteinaceous amino acids are bound together with the help of peptide bond in various manner, then they form the required protein that is necessary for the construction of the body of all type of living organisms. Then another important thing is what? If a pH of a solution is supposed to change, and then the amino acid that is also present in that solution, the property of that amino acid are also supposed to be changes. This is very important. What when a pH of solution is going to change, then the properties of that amino acid that are present in that solution is also supposed to be changes. And when this changes is taking place, then a form is developed. Meanwhile, when the physical and chemical property will change, then what happened? Another important feature of amino acid that I would like to explain is what? The ionizable nature of amino acid. Ionizable nature of amino acid is what a characteristic feature. It is what? When amino acid is supposed to ionize from one form to another form, then during that there is a compound, Zwitterine compound is formed, that you are observing this chart or in this equation. That Zwitterine is what? A kind of compound that is basically neutral. You know what? A Zwitterine compound is have mainly possesses two functional group. One is positive and one is negative. So it overall it becomes neutral, or the overall charge of them is what? Zero. And this is what the Zwitterine form that that is developed when there is ionization of in amino acid is taking place. So guys, this is what a description of amino acid. Then the another type of important organic compound is what? Lipid. The important next important organic compound is what? Lipid. Lipids are what? Lipid or fat, these are generally water insoluble. They may be simple fatty acid or they may be glycerol. Lipids are generally uh, categorized into two categories. One is fatty acid or one is glycerol. And you know, a fatty acid has a carboxyl group that is also attached to an R group. Short before when I explained amino acid, that I told you that in four group, one is R group, that is variable. In the same, the lipids are also supposed to possess one R group, and this R group may be methyl or ethyl, or a series of higher number of CS2. When you're going through with your NCRT, then you will found a example of palmitic acid, a type of fatty acid that contains 16 carbons in them, including carboxyl carbon. Then, guys, the fatty acid maybe could be saturated or unsaturated. What is the mean of saturated, or what is the mean of unsaturated? In 10th class, when you have studied the chapter carbon or their tetravalency nature, that you came to know that if they are saturated, means they are without double bond, or if they are unsaturated, it means they are with one or more double bond, right? Then another type of simple lipid, as I told you earlier, is what? Glycerol. Glycerol is what? A type of a simple lipid, and it is what? It can be classified further as monoglycerides, diglycerides, and triglycerides. Glycerol further categorized as monoglycerides, diglycerides, and triglycerides. Uh, these lipids are generally referred as fats, and generally referred as or classified as fats and oil, and this categorization is based on their melting point. Means oils always have, remember, oils always have lower melting point, and so they remain in oil form even in winters also, right? So it is suggested that the fat or the lipid, this is I'm telling you additional point, that doctor also suggested that the fat or lipid that is liquid in room temperature, that is very much beneficial for health. So guys, oil is what? Having lower melting point even in winter also. So they are beneficial for the health. Now, some lipids or some lipids include one another group in them, and it is what? Phosphorus. So lipid, when they are also possesses phosphorus group in them, then they are referred as phospholipid. And the example of phospholipid is what? Lecithin. The example of phospholipid is what? Lecithin. Then guys, one important thing that I would like to tell you, that living organisms have a number of carbon compounds with some heterocyclic rings can be formed, and these are what that we have observed in two important type of nucleic acid present inside the nucleus of the cell, and these two type of nucleic acids are DNA and RNA, that are further referred as genetic material. DNA, stand for deoxyribonucleic acid, and RNA, stand for ribonucleic acid. Now, the very important thing, now I'm going to explain how these DNA molecules are formed. Earlier we have discussed amino acids, then we have talked about lipids. Now we will talk about the nucleic acid, especially DNA. DNA. Now you are observing a double helicular structure of DNA. In this double helicular structure, you are observing there are two strands. One strand is you are observing that having 3 prime, 5 prime direction, and another strand is anti-parallel to one strand. Means it is having 5 prime to 3 prime direction. I think it is clear to you. You are observing in this diagram. I'm become sidewise to explain the full length diagram. You are observing there are double helicular or spirally arranged two strands, and you will instead, if you will observe the tip of that strand, then you will find one is of 3 prime, 5 prime, and another is of 5 prime, 3 prime. Means 5 dash, 3 dash, or 3 dash, or 5 dash. This is what the anti-polarity of the strand that you will study in detail in class 12th. So this is what the two strands that are spirally arranged, and in between you are observing there are some junction, and in these junction A is connect to T, G is connect to C. What are these ATGC? These are nitrogenous bases. When you going through with your NCERT chapter biomolecule, here it is mentioned that nitrogenous bases are what? Adenine, guanine, cytosine, and thymine. One more nitrogenous base that you will study is what? Uracil. What is uracil? I will explain just after. First, we will try to understand the structure of DNA. So in this structure of DNA, you are observing there are two strands that are anti-parallelly arranged to each other, spirally double helicular structure, and in between there are nitrogenous bases. Now, guys, I'm going inside these structure. When we try to analyze the biochemistry of these structure, then we will found that earlier I told you that when amino acid bound together with the help of peptide bond, then they form polymer of protein. In the same way, DNA molecule is what? The polymer of nucleotide. Remember, DNA is what? The polymer of nucleotide. Now, the question is what? What is nucleotide? Nucleotide is developed from nucleoside. Now the question is what is nucleoside? Now first we will try to understand nucleoside, then nucleotide. So nucleoside is what? I told you that there are four type of nitrogenous bases: ATGC. ATGC means adenine, thymine, guanine, and cytosine. So when uh, with these nitrogenous bases, one sugar group is attached. When with these nitrogenous bases, one sugar group is attached, then the form temporary unit is referred as nucleosides, right? When in these nucleosides, when in these nucleosides, further a phosphate group is joined, then the form unit is further referred as nucleotides, right? So now it is clear what is nucleoside. Nucleoside is simply what? Nitrogenous bases and one sugar group. One pair of nitrogenous base and one sugar group is formed nucleoside, and when in that nucleoside one phosphate group is joined, then they form nucleotide, and when these nucleotides unite together and form a polymer of nucleotide, then what is formed? Then DNA is formed, right? So guys, this is what the structure of DNA. DNA is what? First nucleoside is formed, then nucleotides are formed. One thing that I would like to explain, that I told you one more nitrogenous basis that is uracil. You know what happened? There are two type of nucleic acid. One is DNA, that is very much important, and one is RNA. In RNA, in DNA, what happened? A connected with T, but in RNA, in place of T, there is a placing of another type of nitrogenous base. It is what? Uracil. You just remember what I explained, because the detail of structure of DNA is your class 12 part syllabus. So this is about DNA, RNA, lipids,